This is a diagram of a neuron's node of Ranvier, where action potential takes place. We will demonstrate the 4 steps of this process: resting membrane potential, depolarization, repolarization, and hyperpolarization. On this diagram, you will find the pink triangles represent potassium molecules. They carry a positive charge. The yellow circles represent sodium molecules. They also carry a positive charge. There are 2 types of voltage-gated channels: a sodium channel represented by a blue tube and gate, and a potassium channel represented by a green tube and gate. This is where the structure equals function, because these channels are proteins that recognize and only allow for a specific molecule to come into the cell. The sodium-potassium pump is represented by a purple circle with a brass brand. It has 3 spots for sodium molecules and 2 spots for potassium molecules. Last, in the intercellular space of the axon, we have an orange blob that represents large anions. They carry a negative charge. Let's take a look at the process of action potential. This graph indicates millivolts, 1/millionth of a volt, and time. The number 1 represents the resting membrane potential. Number 2 is the beginning depolarization. 3 is repolarization, and 4, hyperpolarization. This slide represents stage 1, known as a resting membrane potential. This is when a neuron is resting and waiting for stimulation. The charge inside the cell is negative 70 millivolts, while outside of the cell is positively charged. We find that sodium molecules, represented by the yellow circles, are outside the plasma membrane because the ligand-gated sodium channel has not yet opened. Inside the cell, potassium molecules, our pink triangles, are attracted to the negatively charged large anions, the orange blobs. The voltage-gated potassium channel is slightly open, known as a leaky gate, allowing potassium to slowly enter the cell. Depolarization, or stage 2, is the beginning of action potential. The gated sodium channels are now open as a result of stimuli, and sodium molecules begin to diffuse into the cell. As a reminder, positive charges attract negative charges, so the sodium is dying to get in and hang out with those large anions. As sodium rushes in, the inside of the cell becomes more positively charged and eventually reaches positive 30 millivolts. This sudden change in voltage brings us to step 3, known as repolarization. The sodium channel gates become slightly closed or leaky, while the potassium channel opens completely as an additional exit for the ions. Remember, positively charged ions repel each other, so potassium molecules will begin diffusing out of the cell in an effort to get away from the sodium that has now permeated the cell. This diffusion causes the cell to return to a negative charge of minus 90 millivolts. Now, the 4th and final stage, termed hyperpolarization, begins. If you recall, the cell's charge is at minus 90 millivolts, which prompts the sodium-potassium pump to turn on or begin working. The pump has 3 spots for sodium molecules and 2 spots for potassium molecules. ATP fuels the pump to spin, and the cell's bouncer begins to work. Sodium molecules are kicked to the outside of the cell, while potassium molecules are brought in. The sodium channel gate is now fully closed to prevent the molecules from rushing back in, while the potassium channel remains open. The inside of the neuron is restored to a charge of minus 70 millivolts, returning the neuron to a resting membrane potential. The cell is now ready for the next stimulus in the 4 stages to repeat.